So my really good students, they always put the test dates in their calendar. You know, they're very much aware of when those dates are coming up, right? So right now they start going into panic mode because technically it was supposed to be next week. Clearly we're not going to get through all the material in that time, so I moved it this morning. So I just moved it right now here, but I moved it in the computer lab this morning. So it's October 12th, which is a Wednesday. After we come back from fall break, which is October 10th and the 11th, right? We don't have classes. That's fall break. What other thing? So that's number one, ease some minds. The next is, I've finished all the Wiley Plus assignments, but I just posted the first one. I'm going to post the next one probably after class. You should get both of these done by Wednesday. But remember, they are set up so you can do it after the date. Do it as soon as you feel ready that you could take it as if it's a quiz, right? And then do it again, right, to get all your points. Does that make sense to everybody? Okay. So I have all of them done already. I just have to do the like 12 clicks to post it on Canvas. But there it is in our module, right? I even wrote in the title. Right, so that's to cover the first genetics that we've gone over. I've got a genetics part 2. And then today we should hopefully start viruses. So let's finish up genetics part 2. We were on, I know we talked about this. And I think we started to review transformation, the Griffith experiment, right? So his experiment helped prove that DNA was what had the genetic code, the information on how cells make things. So he was able to, when he was trying to develop this vaccine for Streptococcus pneumoniae, demonstrate that it's actually the DNA that tells it how to make a capsule and therefore made that particular strain pathogenic. And so even though these guys were dead, their DNA was picked up by the living cells, and they were able to make the capsule become virulent and kill the poor mouse. So this happens in nature. So it wasn't just because of his experiment, he just happened to discover something that naturally occurs. So this process is now known as transformation. And this is where extracellular DNA, so outside of a cell, can be introduced directly into a cell. It doesn't require direct cell-to-cell contact, but it does require the cells to be what we call competent. And some bacteria are naturally competent, which means they will take up DNA from the environment. They have the mechanism to do this. Others we can cause to become competent in the lab. And so those of you guys taking the lab class, towards the end of the semester, we're going to actually put DNA from jellyfish that's been put into a plasmid, right, so extra circular piece of DNA, into bacteria and make them produce green fluorescent protein and actually glow fluorescent green, where E. coli doesn't normally do that under UV light. The reason why is we're going to give them the DNA on how to make that protein. Okay? So, as I said, we can do it artificially as well. In the lab, we use calcium ions to help facilitate the movement, and we use a special strain of E. coli. It's not the standard stuff you work with. Some labs actually use electroporation. They actually use electronic pulses to push the DNA into the cells. That takes, high-tech equipment that we don't have the need for here at Delgado, so we don't actually have it. Who knows? We might someday, though, with the biotech program. They might start doing electroporation. So competent cells that are naturally competent have receptors on their surface, and under certain conditions, they can attach to external DNA. So a cell's died, their DNA has become part of the environment. This living, competent cell comes along, comes in contact with this DNA and it binds to receptors on the surface of the cell wall. And it's actually going to digest away one of the strands of DNA. It only takes it in single strand. And then the cell isn't truly transformed until it actually finds a place in the chromosome to put that information. So it's more apt to happen between organisms that are somewhat related, because they got to find some place that's somewhat complementary, that's similar, to insert. Now, they're not truly transformed unless they actually incorporate it into the DNA. Sometimes they'll just degrade it. They can't find a place to put it, so it just gets chewed up. Then it's not transformed, right? Because it didn't incorporate that DNA into it. Thought I had another slide for some reason. So a couple of the terms that we use sound very similar, right? We've talked about transposons, right? Transposable elements, those jumping genes. That starts with trans, right? This is transformation. It starts with trans. Then we're going to talk about transduction. Oh my gosh. That's 3 words that start with trans, right? So obviously focus on the second half of the word, right? That's going to help distinguish one from the other. The trans, of course, means that stuff is transferring from one place to another, right? And in this case, since we're talking about genetics, what is that molecule? DNA. Right, so we're talking about DNA moving. So transformation, I always think about form, right? Transform. Even think about transformers, right? Most people are aware of those robot things that turn into cars and then into robots, right? They transform between the two. Right? My son's convinced that that Camaro that somebody's driving around New Orleans is Bumblebee. Trying to explain to him it's not going to turn into a robot. That's make-believe. But who knows, y'all, someday that could really happen, right? Especially the things that we can do. But so think about that transformation, that big change that can happen, right? And DNA is the information that determines what these cells are going to look like. So they can really transform. I mean, they can really look extremely different, like the E. coli that glows green, right? That's a huge transformation. That's a huge change from what it was before. Some of them, of course, are changes that are not as very easily visible, right, like green fluorescent protein. Some of them might be the ability to metabolize something, right? So you'd have to do tests to be able to find out, like we talked about screening for mutants and looking at those types of abilities. Does this make sense to you guys? Right? So think transformation. Think about that ability to change dramatically based on the DNA they receive from the environment. Conjugation, on the other hand, this does require cell-to-cell direct contact via the sex papillus. Pilus. I always want to add another P. Pilus. So a lot of times this happens because they contain a plasmid known as the F plasmid, and this stands for fertility. And so a cell that has this plasmid is said to be F+. And on this plasmid is the information on how to make that sex papillus, to be able to join 2 cells. So an F- doesn't have that. So an F1, an F positive cell that has that ability would join up with one that doesn't. So here they are. So here's the F plasmid. It has the ability to make the sex pilus. And so they're able to join up with each other, literally connect with one another. There's a pore that opens up a channel. And that information, just one strand, is copied and sent through to the minus cell. But of course, once he has it, he has the information, he's now F+, right? You put one F+ E. coli in a test tube with a whole bunch of F-, guess what you end up with? all F pluses, because they just keep giving it to all their friends. Because once this guy becomes a plus, he can give it to a negative one, right? And they just keep doing it until the whole test tube, right? So it's just like a sexually transmitted disease, right? It spreads fast. But you have to have that direct contact. Those cells have to directly hook up with each other. Now, I'm just talking about plasmids, right? Your book goes into a whole lot of detail on how stuff can come off of a chromosome and go on to a chromosome. I want you guys just to have the basic knowledge that conjugation requires the direct cell-to-cell contact, right? The sex papillus is how they join up, and that information goes from one cell to the other. And of course, that cell is now changed that received that information, right? So this is sometimes referred to as bacterial sex, because that's what sex is an exchange of genetic information. And, you know, it's very reminiscent of sex that we think of, too, because it requires direct cell contact. So that DNA is protected, right, because it's going through that connection between the two cells.
Do the cells ever, have they ever monitored it as where the cells are going to pass that on to a positive? Do the two positives stay away from each other and go to the negative?
Right. So the two positives, they have a way of sensing that they already have that information.
No need to connect.
Yeah, they're not going to join up and give them stuff they already have. Yeah. So the next is transduction. So remember I said another trans, right? But now I want you to focus on the second half of the word, duction. So think about a duct, right? Stuff, again, travels through a duct, through a vehicle, through a conduit. So again, the DNA is going to travel through something. Anyone know what it is in this case? What's going to protect the DNA as it moves from one bacteria to another? No? A virus. A virus. So even bacteria get viruses. All living cells can be infected by viruses. So what happens here is that a virus infects a bacteria. And a lot of times what they do is they chop up the bacterial cell's chromosome. So it stops it from being a bacterial cell. Because a virus, what it does is it takes over that cell and says, okay, you're a virus-making factory now. You're going to make viruses. You're no longer a bacteria. So in order to do that, it takes over. Well, one of the ways you can take over is you shut down the command center. Right? So they chop up that DNA. So then it takes over the ribosomes, gets the ribosomes to make its protein coats. It uses its enzymes to copy its nucleic acid and packages all that stuff up, and then it busts out of the bacterial cell. The problem is that rarely, but it happens, instead of viral DNA getting packaged up in that viral protein coat, it packages some of that leftover bacterial chromosome in that viral coat. So now when the virus goes and lands on and goes to infect and inject its DNA into another bacteria, it's not actually viral DNA. It's bacterial DNA from the cell that it infected previously. So that virus is kaput, right? Because it's not going to make viruses anymore. But it did inject DNA from one cell, bacterial cell, into another. Now, the donor here, right, this is the donor that get infected by a virus. So unfortunately, this guy dies, right? Just like with transformation, some bacteria somewhere died, and its DNA became part of the environment. Unlike conjugation, right, they hook up, both of them survive, right? Donor gives to recipient, recipient can become donor, and they just keep passing their information on. Here, the donor dies due to a viral infection. Bacterial phage, or just phage for short, are the name given to viruses that infect bacteria. So here's the bacterium. Here's the phage. It's injecting its DNA in. So it's color-coded. Its enzymes, as I said, is going to chop up the bacteria's chromosome. So that's all the little blue pieces. And then it starts replicating, copying its own DNA, the viral DNA. The next part is assembly, right, where they're going to package the proteins around the nucleic acid, and it's going to bust out of the cell. This guy and this guy, notice they've got blue stuff inside them. That's to represent the bacteria chromosome, not viral. But this virus is not a sentient being. It's not even a cell, right? It doesn't know it has the wrong information. It's just going to do what it always does. It's going to go and infect another cell by injecting its DNA into it. In this case, it's not viral DNA, though. It's bacterial. And if that finds a place to incorporate into the chromosome, that bacteria now has been transduced. Right? DNA from one bacteria has been added into another one. And depending on what that information is, it'll give it some new ability it may not have had before. So the interesting thing is, this happens, these processes happen all the time. And E. coli 0157, that's a pathogenic strain of E. coli, right? One of the reasons why it's so pathogenic is it produces a toxin called the Shiga toxin. How does it know how to make that toxin? Where's that information in its cell? In its DNA. Okay. The Shiga toxin is the same toxin, and that's why it gets this name, Shiga toxin, that's found in the bacteria Shigella. S-H-I-G-A. Shiga. Shigella, which is a bacteria, produces that toxin. And again, that's why that causes such a serious infection. That exact same toxin, there's one strain of E. coli, it produces it. It's the exact same. Right down to the DNA, it is the exact same toxin. So E. coli basically got it from his friend, right? Shigella? How did he get it? We don't know. Was it transformation? Was it transduction? Was it conjugation? We don't know. But it was definitely what we call a lateral move, right? Where it went from one organism to another. And these guys are gram-negative bacteria. They have a lot of similarities, right? They're pretty closely related bacteria. But plain old normal E. coli does not have this toxin. Thank goodness. Because it is evil. So all of these that we've talked about, transformation, transduction, conjugation, these can happen between like E. coli and Shigella, or say Salmonella and some other closely related bacteria. Some of these can even happen between not very closely related bacteria. But these aren't the normal changes, mutations that we're used to in DNA, right? This isn't where it goes from mom to all her progeny, direct descendants, right? That original E. coli 0157 that picked up that Shiga toxin, somehow horizontally or laterally, right? Whether conjugation, transformation, or transduction. Then passed it on vertically to all her offspring, right? So things will move, right, vertically through a population that's directly related to one another's, right? Ancestors. So imagine your family. But then stuff can also make these big hops, right, laterally or horizontally from like Shigella to E. coli via these gene transfer mechanisms. We commonly refer to these as gene transfer, right, where you're going from one bacteria to another, not within the same family tree or lineage. Does that make sense to you guys? Okay. That's it for genetics. God knows that's enough, right? As you're reading through this chapter, they go into a lot of detail, right? If details bog you down, as I said, stick to those animations. Those are nice, good summaries of those last 3 processes we just talked about. I'm only going to test you in the level that we talk about it in lecture. I'm not going to go into those little picky details. And you'll notice that too by the questions that I picked for your Wiley Plus assignments. So the first one's already been posted that goes with the first genetics, and after class, I'll post the next one. And if you came in late, the test is not next week, it's the 12th of October. Yeah, I'll just go on to viruses. So most of viruses that we're going to talk about is in Chapter 5, but some of the specifics in their life cycle is in, I forgot the number, a later chapter. Is it 8? Oh, I don't want to speak wrong. Okay, Andrea's going to look it up for me. Okay, so for Chapter 5, viruses have been around for probably just as long as probably cells have been around, if not maybe before. We don't know for sure. But smallpox, that particular virus, has been linked back to even in Egypt. Right? So mummies have been found with evidence of smallpox. And there was a fear recently of Siberia, parts of that, the permafrost is melting, and there are bodies buried up there, and they're afraid of smallpox getting back out as people are exposed to those bodies that were frozen solid, and so therefore preserved. So kind of crazy things to think about. So as early as the 1800s, they were able to filter something out of the tobacco mosaic, the tobacco plants, what we call today is the tobacco mosaic virus. And so some type of infectious agent that wasn't bacteria, right, that was causing this infection. So Walter Reed, right, you hear that name, right, hospital named after him. But what did he do? He was one of the contributors to discovering that yellow fever was transmitted by a virus, was caused by a virus transmitted by mosquitoes. And we did have that here in Louisiana at one time, yellow fever and malaria. But they had both been eradicated since then from the United States. We, of course, have mosquitoes, right? But thankfully, we don't have the virus, or in the case of malaria, the plasmodium, within the population of people or animals that serve as a reservoir for the virus or the plasmodium, which is great news for us. Not the case for other parts of the world, right? Where these diseases still persist. So what do we mean by virus? What would be a working definition of a virus? And what implications does this definition embody, right? So your definition really is based on your understanding of how these things work, right? And what they're made of. So in the very simplest of things, what is a virus? Is it a cell? No, a lot of scientists describe it as being acellular, which means not a cell. Others would say they're intercellular obligate parasites. So intracellular, which means they're inside of a cell, right? Just like in this picture. Although I have one pet peeve about this picture. I think they're meant to represent size in this picture. Not that, of course, bacteria don't get infected by the tobacco mosaic virus, because that infects the tobacco plant, right? But just meant to represent that all these viruses, and herpes virus, of course, infects humans, that these guys are smaller than a bacteria. All these different viruses. It's not my favorite picture in the new book. They're teeny tiny, between 10 and 100 nanometers. Are you going to see these with a light microscope? No. What type of microscope do you need to probably visualize? Electron microscope, right? Genetically, they're probably a few thousand to 200,000 nucleotides in length. Now notice I said nucleotides, and I didn't say DNA, right? There's a reason for that. Our virus is made out of DNA. Some of them are, some of them are not. Some of them are RNA. So in the case for them, right, even what we're used to for cells, right? A cell has DNA. It's got to have DNA. Well, some viruses, no DNA at all. Just RNA. Most scientists, too, don't consider these living things. I don't. My definition of a living thing is at least a cell. These guys are not a cell, because some of them don't even have DNA. They can't replicate on their own, and then they also can't make proteins on their own, because guess what they don't have that you need to make proteins? What makes proteins in a cell? Well, amino acids are put together, but what puts them together? The ribosomes. Viruses don't have ribosomes. But yet their nucleic acid, usually a protein coat, and sometimes actually what scientists call a membrane now. But again, still no ribosomes. Sometimes not even any DNA. They cannot replicate on their own. That's what we mean by obligate intracellular parasites. They have to get into another cell. They have to get into a cell in order to use that cell's machinery to copy themselves. So how many have said or read to kill a virus? They think they're killing viruses. So when you clean your house, are you killing viruses? Yeah, the label might say that. But how could you kill something that was never alive to begin with? In truth, you're inactivating them or making them non-infectious. Because they do have to have intact structures in order to be able to attach and get into cells. Whatever cell it may be they're infecting, whether it be a bacteria cell, an animal cell, a human cell, an algal cell, a plant cell. There's not a single cell out there, by the way, that's immune to virus infection. All cells have the potential of being attacked by a virus. We know all too well about viral infection, right? But bacteria, they have viruses that infect them. Plants have viruses that infect them. So the most standard definition, as I said, is they're acellular. They're not a cell, right? They're lacking the major components of a cell. Sometimes they don't even have DNA. They never have ribosomes. They can't replicate on their own. They can't respond or metabolize on their own. They're essentially inactive outside of a cell. Inert. Can't do anything. So although you may think you're killing them sometimes, you're really just inactivating them, making them non-infectious. So when we start looking at their morphology, there are some terms that we use. So what's a viron? What are naked viruses versus envelope viruses? What do we mean when we say nucleocapsid? Pentamers, this one kind of gives you a clue in the name, right? Because what does pent mean? 5, right? So this is where there's 5 proteins coming together. Where hexamers have, what's hex? 6, right? So how are these arranged to form capsids where you have helical arrangements versus isohedral, or the more complex, like the bacterial phage we'll look at? So as I alluded to earlier, viruses are either DNA or RNA. They're not both. And then, usually when I say DNA, you think double-stranded or single-stranded. Double-stranded, because that's how DNA is in our cells. Guess what? Some viruses have single-stranded DNA. And RNA, right, we're used to seeing that as single-stranded, right? Some viruses have double-stranded RNA. So it can be single-stranded or double-stranded, DNA or RNA. So a lot of scientists will, specifically when they're talking about a virus, say whether it's single-stranded or double-stranded, and whether it's DNA or RNA. So notice for the polio virus, it's a single-stranded, that's what that SS stands for, RNA molecule is its genome. It infects humans. Unlike the tobacco mosaic virus, this one affects tobacco plant and related plants. It's also single-stranded RNA. T4, the bacterial phage that will infect E. coli, is double-stranded DNA. Smallpox virus that infects humans, double-stranded DNA. So the core of it is its genetic information. Is it DNA, is it RNA, is it single-stranded, or is it double-stranded? Now, of course, they only showed you 2 examples here, double-stranded DNA and single-stranded. But there's double-stranded RNA and there's single-stranded DNA. No matter what its nucleic acid, it's always surrounded in a protein coat. The name we give to that protein coat is the capsid. The capsid is made of capsomere proteins. So individual proteins, and sometimes they're in special groupings, right? The pentamers, where there's 5, or hexamers, where there's 6. I'm trying to remember if I posted something I want to show you guys. I'll look in a minute. Now, of course, the name we give to the nucleic acid plus that protein coat is the nucleocapsid. Nucleo referring to genetic information. Capsid, of course, the protein coat. Some of them, not all, will have an additional layer. We refer to that layer as the envelope. And it is a plasma membrane that surrounds the capsid. So it's the outermost layer if they have it. But it is a modified layer. It gets it from the plasma membrane of the cell it infects. Most animal viruses, the ones that infect humans, are enveloped. They use our membrane to make their envelope. It isn't strictly just our membrane around them, right? But they use our membrane as the building blocks for their envelope. So notice here it says non-enveloped. Some scientists will refer to that too as naked viruses. Because as you can imagine, most of the viruses, especially when we started studying human stuff, had that envelope. So then all of a sudden we're like, oh, there are viruses that don't have an envelope. So they were missing a layer, you could say. So they were quote unquote naked viruses. So non-enveloped and naked means the same thing. So how is that protein packaged around the nucleic acid? Well, it depends on the virus. The tobacco mosaic virus, it looks like these long tubes, right, in this picture, in this electron micrograph. What they have is their RNA is helical, and the protein subunits that make up the capsid connect to it in the same fashion that the nucleic acid is helical. So although it doesn't look like a helical structure, right, when you look at it in this form, if you were to see it in the closer up form, right, it's like a spiral tube that forms. That's why it's called helical, although it doesn't really appear that way, right? Isohedral is where the capsules, I don't know how to describe this one. They end up with equilateral triangles, and they end up with 20 different ones. And so what I will do next week, or next class, Wednesday, is I will actually bring over my models, my three-dimensional models from lab. Those of you guys in lab, you might have seen them hanging on the wall in lab, but I'll actually bring a couple of my other models in. But if you want to fold up and make your own isohedral, I usually post it, the model for you guys. I think I forgot to. Here. I know I have it in here in my files. So this is a virus. It is, did I open the module too? If I can't find it quickly, I'll just do it after class. There it is, virus model. It's a PDF, printable document format. So this is cool. You can print it out in color or black and white, of course. In color, you can really see the different proteins because they've been color-coded. But you'll see right here, see how there's 5 blue? When you cut this out, and you'll see there's triangles, and you fold it along the triangles, and you paste all the sections together, you get that isohedral shape, right? So you'll get a three-dimensional shape. So I'll bring my models next time so you guys can see that. All right, so you just print out this page and you cut it out and fold it along all the triangles, and then you can tape it together. So those are the 2 most common shapes that you're going to see, the helical and the isohedral, for the proteins that protect the genetic code. You can see more irregular, right? So they don't have a regular symmetry to them, or even much more complex. So this is an example, right, of, it looks, I don't even know how to describe the shape. You know, you see the capsid proteins on the inside here, and here's the DNA wrapped around it. And then so each of these is represented a little strand, so and then the polymerase attaches and copies that information, but it's wrapped around these capsid proteins. It's an interesting shape. This one's my favorite one. This is what most of the phage, the bacterial phage that infect bacteria, look like. So really complex, right, arrangement. Almost looks like a little spaceship. And they literally land on the surface of bacteria, almost like little spaceships. There's YouTube videos and everything that show. But so these proteins here literally land on and attach to receptors on the bacterial cell surface. And then this tail portion will actually compress down and sit right on the bacterial cell. And then up here in this capsid portion is where the nucleic acid is. And it literally injects itself, shoots it through into the bacteria cell. So this whole big apparatus doesn't go in, just the nucleic acid and any enzymes that are in this capsid portion. They're pretty amazing looking little things. So as I mentioned earlier, ones that don't have the envelope are sometimes referred to as being naked. And both these viruses, overall, most of the animal viruses look spherical because that envelope layer causes them to be spherical. But inside, whatever the capsid is that's surrounding the nucleic acid can vary in shape. You can't really see it all that well in these pictures. These two are just showing you that they have envelopes, both influenza and HIV. So we're going to go into details in the later chapter of the reproductive cycle of a virus. But you guys should be able to list generally in order the steps, right? So the very first step for all viruses, chapter 8, look at that. It's right there. Is attachment or adhering to the surface. It's literally binding to the surface of that cell. Some receptor that that cell has that it's using for some other purpose other than for a virus to land on them is there. And viruses have adapted to literally attach to these cell receptors. So this is why when we talk about HIV, it doesn't infect all your cells, does it? Anyone know what cell type it infects? It affects a cell of your immune system, but a specific set of cells. Even more specific than white blood cells. Even more specific than just one type of T cell, because you have two different types of T cells. Your helper T cells, because your helper T cells have a cell receptor on them known as CD4. And that's what HIV binds to, is that specific cell receptor on the surface of your helper T cells. And that's why it affects that component of your immune system. Unlike measles, right? When you get measles, it's pretty much widespread, right? So the cell receptor it's attaching to is one that's very common amongst most of our cells. Unlike polio, right? Polio only affects certain cell types. Again, that's because that receptor is only found on those cell types that it's attaching to. So that's number one important, and really important to how we can protect ourselves against viruses, right? Because if we can stop them from doing this, can they get in? No, the first step, they must attach. They must adhere. That's crucial for all infections, even bacteria, right? They've got to be able to adhere to us first, right? And then the rest is makes sense, right? It's got to get in. It's got to enter somehow. It's got to replicate somehow inside. It's got to assemble. And then it's got to exit. Right? In the simplest terms, attach, enter, replicate, assemble, exit. Right? You can use other adjectives for these things like synthesis. Right? We know what that means, right? To make the pieces. Make the DNA or the RNA. Make the protein. So we'll pick up here next time, and I promise, before 5 o'clock in the morning on Wednesday, post the next virus lecture. Because there is a part 2 to go at chapter 8.